Hey everybody. So I started doing this drawing just last semester, and my students said that they really liked it and it worked for them, but of course it was face to face. So while I planned to do it the next time I taught this class, I didn't know I was going to be doing it online. So I'm going to try and do a drawing with you to explain operons and see if maybe we can still have a little bit of that one-to-one, face-to-face feeling, that connection. So when we're talking about operons, we're talking about all the genes of a particular function organized in one area on DNA. So that's my DNA, that's my double-stranded DNA. And this gene here, this part might be transcribed into and then translated into an enzyme that's required for bringing sugar into the cell, and this one might be responsible for breaking the sugar down into monomers, and this one might be part of converting that sugar into glucose so that it can go into glycolysis. So several genes on the same segment of DNA, that's called an operon, and it's all controlled by a certain section that's called the promoter. So this is gene 1, and gene 2, gene 3, and there can be several. And then there's this place up here, and it's called the promoter. And the promoter is the part where the RNA polymerase, which is the enzyme that's going to make the messenger RNA, this would be mRNA, right? And then eventually it would go to the ribosomes and it would make protein, and I just have the primary structure of proteins kind of represented there. So RNA polymerase, polymerase, is what is going to transcribe all of these genes. So it's going to bind to the promoter, because that's the start place, and then it's going to go along and transcribe all of those genes. And what I've drawn here, if this is the normal state, we would call this constitutive, ooh, it really blew into the mic there, sorry, constitutive expression, meaning it's always being transcribed and translated. So this would be what we would call a repressible operon. In a repressible operon, transcription is always happening. We have transcription. So that means that this operon is what we would say is on. And in order to repress this operon, in order to stop it from transcribing, perhaps, if these are genes for bringing in a sugar and converting a sugar to be used, perhaps the cell isn't around that sugar. So why would a bacterium waste the cellular energy to transcribe and translate all of this material if the sugar that it's supposed to work on isn't even available? That's the nice part about operons, is that bacteria can be very energy efficient. They can really conserve energy by turning on and off operons. So what we want is we want to turn off this operon or repress it. And the way that you can repress an operon, this would be my after, is if we take a molecule and we bind it to what's called the repressor. Now, up here I'm going to draw the repressor up here. Up here, the repressor is inactive. It's an inactive repressor. And down here, notice the repressor has changed its shape. It's changed its shape so that it actually fits over top of the DNA, and it's blocking the promoter. And if it's blocking the promoter, that means that RNA polymerase cannot transcribe and translate. So, well, it cannot transcribe, so nothing to be translated, and so transcription is off. Transcription is off. Now, how do we change the shape of the repressor to activate it? This is an active repressor. We change the shape of it by binding it to a molecule. So the nice example for this is I was talking about sugars up here, but maybe these are all enzymes needed to make a particular amino acid. A lot of our repressible operons are ones that make amino acids because the cell needs amino acids almost all the time because it's almost always making new proteins or enzymes for metabolism, cell repair, cell synthesis, things like that. So let's say this enzyme makes part of the amino acid, this enzyme adds, the carboxyl group, this enzyme adds the R group, and so it's an assembly line. And you end up with all of this amino acid, lots of it, so much amino acid. And eventually, there's so much amino acid, we no longer need to keep running this operon. We need to turn it off. And so what can happen is that particular molecule can bind to the repressor. And if it binds to the repressor, remember the three-dimensional shape of a protein, like a repressor protein, is dictated by hydrophobic interactions, hydrophilic interactions, hydrophobic, ionic bonds. And so if you bind it to another molecule, it's going to change all of those interactions, and we call that a conformational change because it's physically changing its shape. And now, when it's bound to what we call the co-repressor, when it's bound to the co-repressor, then it changes shape and it becomes an active repressor, and we turn off transcription. That's a repressible operon. There's also what we call an inducible operon. Now, an inducible operon is going to naturally have no transcriptions. I need to fix this. No transcription, off. Right? So an inducible is naturally no transcription or off. Why is it off? Because the RNA polymerase cannot bind to the promoter. That means the repressor is active. So in an inducible operon, the normal state has an active repressor. No transcription. So notice, when the repressor is active, we have no transcription in both cases. But in this case, this is more my sugar example, because this would be like, okay, the cell, can use fancy sugar X, but it's not available, and then all of a sudden, you eat a burrito, and your gut bacteria now are exposed to fancy sugar X, and so they want to turn on that operon. So the way that we're going to inactivate the repressor is by something that we call, let's see, I'll use pink. So all of a sudden that sugar molecule is available. Fancy disaccharide sugar. Those can bind to the repressor, and when they bind to the repressor, they're going to inactivate it. Inactivate repressor. Now the repressor will no longer fit over top of the promoter, which means that RNA polymerase can come along. Oh, I did those in the wrong color. There's an inactivate repressor. RNA polymerase can come along. Polymerase, short for polymerase. And it can transcribe all the genes needed to use that sugar. And then they'll eventually be translated into protein and folded into their tertiary structures. When this molecule causes induction of transcription, it induces transcription. So think about probably the only other time you really hear the word induce, you think about induce labor, right? Like you start labor, you turn on labor. So transcription is induced, it gets turned on. This molecule that inactivates the repressor, which allows that operon to be induced, is called the inducer molecule. All my names end in ER, so I try and put R on things. Inducer molecule. So, in our operons, we have an inducible operon, which is off and then gets turned on, or induced. We have a repressible operon, which is constitutively on and then can be repressed. Oops, I'm messing with my light. Anytime there is no transcription, the repressor is active. Active repressor, active repressor. The way we activate a repressor in a repressible operon is with a co-repressor. Co-repressor activates the repressor. The way we inactivate the repressor in an inducible operon is with the inducer molecule. So, take a look at your sheets that you drew along with me. In which operon is transcription usually happening? Repressible. Right, constitutive. In which operon is the repressor usually active? Inducible operon. Let's see. In which operon is the promoter usually blocked? Promoter usually blocked. The inducible operon. How do we induce transcription in an inducible operon? We have to inactivate the repressor. How do you inactivate the repressor in an inducible operon? Exposure to the inducer molecule, and then we get transcription. In a repressible operon, we usually have constitutive transcription. It's usually on. So is the repressor usually active or inactive if transcription is happening? It's inactive. How do we activate the repressor in a repressible operon? By exposure to the co-repressor. Get that conformational change and we turn off transcription. So, if transcription is usually happening, or if transcription is happening, period, is the repressor active or inactive? If transcription is happening, the repressor is inactive. Inactive repressor. Okay, I hope that helps. I'm going to close it, and you can practice.